So we're talking about meiosis, mitosis, cell division, and the whole purpose of this is to split these things and to copy the DNA to give it to our daughter cells. All right, and so first we've got to talk about DNA structure. DNA structure. Can you guys see this pen from the back?
Yeah.
Good. Yes. So I've got DNA structure. There's a couple different terms we've got to talk about. The first one we've got to talk about is called chromatin. Are you looking for a stapler, Amy?
Yeah.
On the bookshelf right there.
Thank you.
All right. Chromatin looks like what? It's all uncoiled DNA. We said it kind of resembles spaghetti in a picture. So this is uncoiled DNA. All right. When do I see this? Yeah. Seen during interphase. Okay. So all this chromatin, this uncoiled DNA, is seen during interphase. And then I've got a couple other terms. I've got chromosome. Oh, and the whole reason why it's seen during interphase is because during interphase there's a big purpose. What's that big purpose? To replicate the DNA. All right, and so let's say it's like I need to make a copy of a book. How can I make a copy of that book if I don't even open the book up? So I've got to open up this DNA or uncoil the DNA so that way I can read it before I make a copy of it. That's why it gets all uncoiled, is to chromatin. Then I've got chromosome. Okay, and so this is going to be tightly coiled DNA. And we said this tightly coiled DNA, it's like your Christmas lights. Come January, when you go to put your Christmas lights away, we said hopefully you don't just take your Christmas lights, ball them up, and then throw them in a big bin. What do we say you should do? Okay, you should get something, they have little things that you can wrap your Christmas lights around. We said that's analogous to a protein. Because your DNA, when it's tightly coiled up around these proteins called histones. So these chromosomes are tightly coiled up DNA and protein. All right? And it takes two forms. We can see it in the shape like this, so it's in the shape of an X, or when it looks like the single turd. Okay? Those are the two structures you might see a chromosome looking like. If it is like this, what do you know is it's undergone?
Interphase.
It's undergone interphase because it's duplicated, and now this is two of these attached together at the center. Each of them have a centromere. This is their centromere. Two of these attached together at their centromere forms this. This is after interphase. This is going to be after anaphase, okay, or during anaphase and after. All right, and then I'm left with one last thing called chromatid. So I've got chromatid, and do you remember what chromatid is?
One half.
It's one what? It's one half of a replicated DNA. All right, so if I were to look at it and it's like, that's a big one like that. If I were to take it, that is one sister chromatid. Okay, that's sister chromatid right here. And so for the sake of this, just between the two, I've got mitosis, all right? And what's the ultimate goal? Is to produce how many daughter cells?
2.
I want to make 2 daughter cells. That are what? Are they, what the heck did I write? I wrote daughter cells. Cells. Do I want those daughter cells to be the same or different?
Same.
That are identical. All right, so if I look at this and I say, here's my first cell. Mitosis, the cells that I start with, are they haploid or diploid? Say diploid, say diploid, say diploid. Diploid, thanks guys. All right, if I look at it, 2n equals, in humans, 46. Okay, 2n is going to mean I have 2 sets of chromosomes that's going to total up for 46 of these. My end product is to produce how many daughter cells? 2. So, and we want them to be identical. So if I look at my two daughter cells, I'm going to produce two daughter cells that are also diploid. Cool. So that's my goal, is to produce two daughter cells that are diploid. So this is going to be for growth, repair, and replacement. I need diploid cells. This is going to happen in all my body cells except for my gametes, which are sperm and eggs. That's the goal. Okay? Growth, repair, and replacement. It's like a Jack Johnson song with 3 R's. Reduce, reuse, recycle.
That's a G.
I know, that's what I'm saying. It's like the Jack Johnson, like 3 R's. Reduce, reuse, recycle.
But it's a G.
Okay, we can just call it GER.
That's still a G.
I know. Never mind. School is fine. Okay, interface.
Wait.
All right, interface is composed of how many parts?
3.
I've got G1, I've got S, and I've got G2. What happens during G1? What? Replacement. Okay. What happens during G1?
Growth.
So growth, proteins, and organelle duplicate. Okay? The same thing happens here. Growth. All right, and I'm just going to put parentheses. That means the same thing as this. I just don't want to write that. But then something happens during this S phase. What happens there? Replication. So DNA replicates. Okay, this is where the DNA is duplicated. It's during this S phase. That's why my DNA had to uncoil, so that way this right here could look at it and it could replicate this. So let's say I'm looking at some cells. And you'll see a term tomorrow, that's why I'm introducing it right now. You'll see a term called assay. It means basically to measure the amount of DNA that's in there, and they measure it in like picograms. So it's essentially calculating what is the mass of DNA inside of a cell. Let's say I've got 2 cells. Okay? I've got, here's cell 1, and let's say I've got, I'm just gonna put 20 picograms right here. And let's say I've got cell 2, and this one has 100 picograms. Okay? So if I'm looking at these things right here, do you think both of these cells would take the same amount of time to undergo interphase?
No, because they have to rip.
But they're both cells.
100 picograms takes more time to replicate the DNA.
Why?
Because there's more DNA to replicate.
Okay, so someone's making the argument that this cell's going to take longer because it's got more. So if I were to look at the 3 stages of interphase, which stage of interphase is it going to spend more time in?
S.
The S phase. Let's say if I were to kind of make this analogous to me doing something, let's say I want to make a project where I'm going to have you do this 2 days in a row. I'm going to have you walk to the library and get a book. You're going to bring that book back here, and you're going to transcribe that whole book down onto a piece of paper. And then once you're done, you have to walk to the library, return the book, and come back. G1 would be like you going there. G2 would be like you going back and returning it. S phase would be like you making a copy of that book. If you get a book that's 200 pages, and you had to copy all that down, it might take you, let's say, 4 hours. It'll take a lot longer probably, but let's say it takes 4 hours. If you get a book that is 100 pages, or 1,000 pages, sorry, we went from 200 pages, we said 4 hours. If you have 1,000 pages, is it going to take you 4 hours just like the 200-page book did? No. It might take you closer to 20 hours if you're copying at that same rate. So that cell is going to spend about 5 times as long during that S phase because it has a lot more DNA to copy. It's got to do a lot more work, okay? So that's what's going on there. I just needed to introduce you with that term assay because you'll see that term tomorrow. It means to weigh it. That was a good spot to do it.
Wait,
A-S-S-A-Y.
S-S-A-Y.
A-S-S-A-Y. That's the term assay, means to like calculate out the amount of weight of DNA.
S-S-A-Y. A-S-S-A-Y.
Yes.
What does that do again?
All right. So within this whole process, I've got something big that happens here. I've got a checkpoint. I've got a checkpoint here, and I've got a checkpoint here. I've also got one in mitosis. Do you remember what these checkpoints do? their big purpose is to just make sure, does it have all the proteins it needs? Does it have all the DNA? Does it have the right amount of chromosomes? Does it have all the right organelle? Does it have everything I need? If it does, we can go on. One of these checkpoints is the most important. That is this one right here. So I'm going to write important. Okay? So that one right there is the important one, because if it generally clears the G1 checkpoint, it's usually good to go through the entire cell cycle. So that's the big one. If we were in these checkpoints and it notices that something is wrong, so it says, hey, something's not right here. I don't want you to keep dividing. Do you remember what it does to that cell where it sends it?
G0 phase.
That G0 phase or that G0 phase. All right. If things are messed up, it sends it to that G0 phase. All right. Which is going to be no division. All right. We have some cells in our body that are permanently in that stage. Do you remember what they are? Certain muscle cells, like our cardiac muscle cells,
Nerve
and then nerve cells in our central nervous system. They don't divide. They're permanently in that G0 stage. All right. They get up to that point where they're mature, and then they're done, and they stay there. Okay. And so they're stuck in that G0 phase. They're not dividing. That's why if you harm them, you're up a creek without a paddle. Okay. So I underwent that. Prophase. I'm going to sum up prophase and prometaphase. On your test, there's no difference between prophase and prometaphase. It's all summed up as prophase. But I needed you to be familiar with that because on the AP exam, they may throw out prometaphase at you. Okay, so prophase. Do you remember what happens there? The nuclear membrane starts to dissolve. I have my centrioles that have formed. I've got my chromosomes in here. Something big happens. In interphase, what was the DNA? The DNA was in which structure?
Chromatin.
In chromatin. During prophase, chromatin condense to form chromosomes. Okay? And then spindle fibers form. And nuclear envelope or nuclear membrane dissolves. So I go, what's the next stage? Metaphase. Okay. So in metaphase, what happens?
The chromosomes line up.
Those chromosomes are going to line up right along that metaphase. I'll put 3 instead of just 2 in there. There we go. Okay, so those chromosomes line up on that metaphase plate because the spindle fibers push them. So spindle fibers move C-somes. They're going to move the chromosomes to the metaphase plate. Okay. What's the next stage? Anaphase. What happens there?
Pulled apart.
Ooh, there we go. Heard it. I was looking for that kicker. Sister chromatids get separated. Okay. So spindle fibers shorten. All right. It'd kind of be like if I had fishing line and I start reeling in that fishing line. That fishing line is shortening. All right. So I'm going to start breaking down the spindle fibers and I'm going to shorten it and shorten it and shorten it. And in doing so, it's going to pull those sister chromatids apart. So spindle fibers shorten. And sister chromatid are separated. Okay. Then I go over here. Telophase. All right, in telophase. nuclear membrane begins to reform. And DNA begins to uncoil. And then finally, cytokinesis. So technically, if I were to look at it, this is not mitosis. Mitosis is prophase, metaphase, anaphase, telophase. And then that's the end of it. And then I go to cytokinesis. And in cytokinesis, I'm left with this. And my DNA is in the shape of chromatin. Because now I'm back at interphase. All right, so I get two identical daughter cells. Okay, cool. Let's say I look at this. This is where stuff's gonna get tricky. Okay, let's say I go G1, S, G2, M, and C. This is my entire cell cycle. All right, so that's the entire stage of the cell cycle. Let's say I do this assay again and I calculate how much DNA is in there. Let's say at the G1 stage, I start out with 20 picograms of DNA. At the end, so that's where, when I measure it right here, that's where I'm at. When I'm right here, how much DNA would I have?
40.
I'd have 40. Why?
Because you were
Because I
replicating.
duplicated it. I'd have 40. When I go to measure right here, how much do I have?
40.
At the end of G2, I still have 40. Here's where it's going to get interesting. So right here, how much do I have? How many cells do I have here? It's still 40. What about at the end of cytokinesis? Now I have 20. That's what I was saying. It's going to get tricky because technically I have not divided the cytoplasm yet. I still technically have one cell. After cytokinesis, now I'm back to 20. So if I look at my DNA amount, it's going to climb right here during synthesis, and then it's going to go, and then during cytokinesis, it's going to go down. All right, so during meiosis, I'm going to eventually split it to cytokinesis where it goes down. Okay? One more little trick question or question to think about. Let's say all the cells in our body, when all our cells undergo one round of division, all the cells divide. Let's say I start out with 2 cells, okay? I start out with cell 1 and cell 2, okay? And we say every 24 hours my cells undergo a round of division. So if I ask, how many total cells do I have after 72 hours? How many rounds of division did I undergo? If they divide every 24 hours, how many rounds of division did they undergo? 3. 72 divided by 24 is 3. How many cells would I have at the end of 24 hours, or at the end of 72 hours? After 3 rounds of division.
8.
What'd you say?
It's 8. 2 3 parts.
Round 1. That's round 1. Round 2. Round 3. I'm left with 16. Okay? I'm left with 16 because every cell duplicates in that round. And every cell duplicates, and every cell duplicates. So if I took it out to 4 rounds, how many would I have? 32. 5 rounds, 64. 6 rounds, 128. Okay, so that's what's going on there with all this division. So there's, have you met Ted? There's mitosis. I don't know if you need that for anything. It also is filming, or if you want to look at it, or just sit back and think about it and reflect. I'm still trying to figure that out. What? Nothing. I was going to be really mean there, but I'm not going to because she's actually a nice girl.
What are you gonna say?
Nothing.
I don't know. Tell me.
No.
Why not?
Because it was ultra mean. It wasn't true, but I was just going to be ultra mean there for a second.
Come here and tell me.
Because you said, what are you looking at? I was going to say, I don't know. I'm trying to figure out what that sorry, pathetic excuse for a face is attached to your neck.
That's not even me. It's funny. What are yours? There is? Wait, Trads.
What?
Are you having a study for the studies?
Fourth period? No, I have a meeting. What time is this class out? Huh? 39? Oh, yeah, we got time. That was an M for Monica. That better? Okay, sorry. All right. So I've got these checkpoints, I've got all this stuff, but sometimes if the checkpoint, so what these checkpoints are for is to make sure everything's okay. But let's say I'm a cell, I'm dividing, I divide, I divide, I divide, I divide, I divide, I divide, I divide. And eventually I get right up next to a cell, and it's called density dependency. And I've got receptors on the outside of my cell that are going to say, hey, I'm right next to another cell. You should stop dividing me. And it sends that into the cell, and the cell says, all right, cut it out. No more dividing. Everything's cool. But sometimes I've got some mistakes that have happened. I could have an extra chromosome in there that's sending mixed signals. I could have had a mutation that caused a problem with my DNA, and now my DNA is causing different proteins to be made, and so it's a mutation. And so now these cells don't know to stop dividing, and they keep dividing, and they keep dividing, and they keep dividing. And what does that result in?
Cancer.
It results in cancer. Okay? All right, and it's unregulated cell growth. Okay. Now that we're going into, so now we're going to step into mitosis, meiosis, but we got to talk about haploid and diploid and stuff. So I've got this term haploid. If an organism is haploid, how many copies of each chromosome does it have? One. So I represent that by saying just N equals and then the number of chromosomes. Let's say for this organism I'm talking about, I'm going to say N equals 25. All right, so it's going to have one copy. Oh, I don't know what was going on there. One copy of each chromosome. Okay? Then I can go into diploid. All right? Diploid has how many copies? Two copies. All right? So I've got two copies of each chromosome. And so if N equals 25, what would 2N equal? 2N would equal 50. So if I said an organism 2N equals 50, how many pairs do I have? I've got 25 pairs of 2, because remember I have 2 copies of each chromosome. So I've got 25 pairs of 2. I can also have like a triploid organism. So you get into like plants and stuff, because when plants were evolving, they said, you know, I really just want to make AP Bio students miserable, and freshman kids and kids that are taking botany. So we've got plants that can be triploid. All right, and these things have how many copies? 3. So if we said N equals 25, 2N equals 50, what do you think a triploid organism would look like? 3N equals 75. Okay, that's the whole goal there. Okay, cool. Now I get to meiosis. In meiosis, I start off with a diploid cell. I'm going to start off with a cell, let's say in humans, 2N equals 46. And then I split, okay? And I go to right here, N equals 23. Because remember, after telophase 1, those cells are now haploid. I separated the homologous chromosomes. Am I done with meiosis now? Nope. I've got to undergo one more round of division. Okay? So here it is. Let's say I've got this right here, and I put another one. Interface. Okay? If I look at this, let's say I weigh the, or I say the DNA again, and I look at it, and let's say right here, I've got 20 grams, 20 picograms. Okay? I don't even remember how to draw a symbol for pico. Let's say I've got 20 picograms. After interface, how much DNA should I have? Someone said it. Starts with 4, ends with a D. 40. Okay, thank you. All right. So I've got 40 picograms right here. After I undergo meiosis 1, how many does each cell have? 20. What about here? How much does each cell have? 10. Okay, so if I'm talking about the amount of DNA, the weight of DNA, if I were to say, compared to the cell before interphase, how much DNA do the daughter cell, or how many daughter cells have, and how much DNA does it have compared to the cell before interphase? How many daughter cells do I produce? 4. Compared to the cell before interphase, how much DNA does it have compared to that cell before interphase? That's half the amount. What if I say the end product of meiosis, how many daughter cells and how much DNA does it have compared to the cell after interphase? It has 4 daughter cells and 1/4 the amount of DNA. Okay? So when we generally associate meiosis, we say, oh, okay, I'm going to produce daughter cells that have half the DNA. Because we talk about it right here. But if you ever got a question that says comparative to the cell at the start of meiosis, how much DNA does the daughter cells have? You've got to figure out, does it want to know before or after interphase am I talking about? Okay, so it'll usually tell you. So if it's before, I've got half the amount. If it's after interphase, and I'm talking about relative amount of DNA, it's actually got 1/4 the amount of DNA. Okay, or 1/4 amount of DNA, because I had to double that DNA before I underwent this. If I didn't double it, and I just went from here and went 20 to 10 to 5, okay, which would still be 1/4. Okay, cool. All right, any questions yet before we get into the stages?
Wait. Would you say so at the end, 4 daughter cells are produced, and 1/4 amount of the DNA was before?
It has 1/4 of the amount of DNA compared to the cell after interphase.
Okay, so the 1/4 of the amount of DNA was before interphase? Or what?
After interphase.
Oh, after.
Half the amount compared to the cell before interphase. And that's if I'm talking gram per gram. Okay, let's talk stages. So in meiosis, I start out with prophase 1. All right, and how this stage looks right here is similar thing's gonna happen. All right, but in this, all right, how is this different than prophase of mitosis?
You're in tetrad.
Okay, they're in tetrad. So in this stage, chromatin condense. All right, and it's going to condense to form these chromosomes. Homologous pairs. These homologous pairs form. All right, and this is called a tetrad. The process of this forming is called synapsis. All right, and it's called a tetrad. When it does this, something massive takes place. All right, what is that process that takes place when it forms this tetrad? Crossing over takes place. All right, and so it's going to exchange small segments of DNA. Do you remember what those small segments are called? It was on your little Poggle activity. Those segments that are swapped. They were called, you'll either hear it called chiasma, or like you might hear it chiasma or chiasmata. Okay?
Yeah, that's the point where they cross over.
For what?
For the crossing over. That's what it said. Like it was the point at which they like crossed over and went to the other side.
Oh yeah, that's what it's going to ask you. Yeah. Okay. So crossing over is going to take place. This is going to be the parts that swap out, or the point at where it swaps out, the little pieces. Okay. The segments that get interchanged. So that's what happens during prophase 1. This is a very, very, very big step. All right. What? All right. Get your life together, Scalise. Then I've got metaphase 1. All right. During metaphase 1, what happens?
Tetrads line up.
Okay. I get those tetrads line up. So if I were to look at this, they line up along that metaphase plate. All right. So they line up right there, and they're still in their homologous pairs. So tetrads line up at metaphase plate. There's a big thing that happens during this step that increases the amount of genetic variation. You see right here, let's say blue was dad, let's say green was mom. Dad lined up on the left for chromosome 1, mom lined up on the right for chromosome 1. Right here, they're different. They had no effect on each other whatsoever, so it's during this stage that a process happens. You remember what that's called?
Independent assortment.
Independent assortment. All right, independent assortment takes place, and that's going to increase genetic variation. Then I go to my next step, anaphase 1. All right, during this step right here, homologous chromosomes are separated. All right. So if I look at it, I'm not splitting these things in half. What I'm actually doing is I'm going to separate the entire X's from each other. All right, so I'm ripping them apart from their buddy. And so now I'm separating these homologous chromosomes. This is the entire purpose right here of meiosis 1, is to separate homologous chromosomes. Meiosis 1's goal is to separate homologous chromosomes. Meiosis 2 goal is to separate sister chromatid. All right. So I separate these homologous chromosomes by the same process. I shorten the microtubule or the spindle fiber, and it pulls these things apart. All right, and then I get to telophase. Telophase 1, and it's going to look similar matter. Nuclear membrane starts to reform, and inside of there, I've got this. Okay, I've got that inside of there. So now these things are going to be haploid at the end of telophase 1. Okay? If I'm looking at these stages right here, of these, so let's say I've got prophase, metaphase, anaphase, telophase. Of these stages, how many of these stages have tetrad in them? Only 2 of them. Tetrad are observed here, tetrad are observed here, tetrad are observed here, tetrad are observed here. How many of these stages do sister chromatids get separated?
0.
None of them. Sister chromatid don't get separated in meiosis 1. Homologous chromosomes get separated in meiosis 1. The whole process of meiosis 2, all right, since we're almost out of here, the whole process of meiosis 2 is all about separating sister chromatid now. So drawing-wise, meiosis 2 looks identical to mitosis. Now I'm separating sister chromatid. So I'm going to take these cells, I'm going to split them during cytokinesis, and now this is the cell that I start with, and I make 2 daughter cells. This is the cell that I start with, I make 2 daughter cells. Each of them is going to split, and those sister chromatids are going to go separate ways. Any preguntas? Okay. Like I said, I'll be here after school. It's a little chaotic in here after school. I will also be here at 6:30. You can also email me if you have any questions.
6:30?
Yes. I have to go